So good morning to all. Today we are going to discuss the development of frog. So in the last class we have discussed about the development of human being. So like that there is another concept, that is development of frog. So how the developmental process takes place in the frog. So already you know the definition of developmental biology. Development of organism from egg to an adult. So that is called a developmental biology. So in the frog, how the adult frog can be developed from the egg, that we have to study. So let's start the structure of egg. From the egg only we have to start. So this is the in the right side corner we are showing the structure of the egg. So there are different parts of the egg, that is cytoplasm, that is pigmented cytoplasm of animal pole. So on the upper side there is a animal pole, and the lower side that is a vegetal pole. And another part is plasma membrane, and the cortex, yolk core, and mRNA, that is maternal mRNA, because the egg is the female gamete. So let's see the structure. So the egg is a female gamete, and it is also called as ovum. So because it is a ovum, or the egg we can call, and it is a female gamete, and it is haploid in nature. Then it is produced by the ovary. The egg or the ovum is produced by the ovary, and the egg is non-cleidoic. Non-cleidoic means the egg is not covered by a calcareous shell. So if the egg is covered by a calcareous shell, then it is called as cleidoic egg. If the egg is not covered by a calcareous shell, then it is called as non-cleidoic egg, as the shell is completely absent. And it is a telolestal, means in the last class also we have discussed the types of eggs based on the distribution of yolk. So here telolestal means the yolk is distributed at the one end of the egg. Only one end the yolk is distributed, hence it is called as telolestal egg, and it is also called as mesolestal. Then the egg contains large amount of yolk, hence it is called as megalestal also. Megalestal egg, because the large amount of yolk is present in the egg. So it is spherical in shape, means the egg is spherical shaped structure, or spherical in shape. Then the egg is surrounded by 3 egg membranes. There are 3 membranes which covers the egg, namely inner plasma membrane, and a middle vitelline membrane, and an outer jelly coat. So these are the 3 membranes which are the membranes which covers the egg, or the egg is surrounded by these 3 membranes. So the egg cytoplasm is called ooplasm. Ooplasm means the cytoplasm of the egg is called as ooplasm. Then it has 2 regions. In the cytoplasm it has 2 regions, namely peripheral cortex and the central endoplasm. Central endoplasm. So the cortex contains 2 types of granules, that is 2 types of granules are present in the cortex. So they are dark brown pigment granules, and another one is cortical granules. And cortical granules are arranged in layer close to the plasma membrane. Close to the plasma membrane. And the endoplasm contains nucleus on one side, and the numerous white plate-like yolk platelets on the other side. So the egg has 2 distinct colors. One side it is black in color, this side is called as animal pole. So already you are seeing in the diagram, that is black color, somewhat dark color structure, it is present in the animal pole, and the nucleus is situated in this side. And the opposite to the animal pole, the opposite side is yellow in color, and this is because of presence of yolk. This side is called as vegetal pole. Then 60% of protein and 40% of fat is present in the yolk. So in the diagram you are seeing the presence of yolk, that is yellow color structure in the vegetal pole. So this is the structure of egg. So just once again we are going to revise, that is, so egg is a spherical in shape, and it is a telolithal as well as mesolithal, and it is a where it is released from the ovary, and it is enclosed by three membranes. Outer membrane is called as Karither plasma membrane, and middle membrane is wetline membrane, and the third is jellicoat. And there are two poles, animal pole and vegetal pole. In the animal pole it is a dark and nucleus is present, and in the vegetal poles it is yellow in color because due to the presence of yolk. So this is the structure of the egg. If asked for 5 marks, you have to write this explanation and draw the diagrams. So next one is the fertilization. So after the union of gametes, the fertilization takes place. Haploid male gamete and haploid female gamete combine together to form the zygote, that is called as fertilization. Or the fusion of sperm with the egg, that resulting in the formation of the zygote. So it is called as fertilization. So fertilization in a frog is the external, because it occurs in the water. That's why it is a external. If it is taking place in the body, or the fertilization takes place in the body, then it is called as internal fertilization. But in the frog it is a external fertilization. Then so it is monospermy, that is only one sperm fuses with the egg. Only one sperm fuses with the egg, hence it is a monospermy. So the sperm enters by animal pole, dissolving the plasma and vitelline membrane. So from where the sperms are entering, from which side means the sperms are entering by the animal pole by dissolving the plasma membrane as well as the vitelline membrane. So at the point of entry, the cone of reception is formed. So where the sperm is going to be enter, so that point of entry is called as cone of reception, that is formed. So penetration path is also going to be formed, and copulation path by sperm head to nucleus is also formed. So after the sperm entry, the second polar body is going to be released, and egg changes into the ovum. So after the entry of the sperm only, the second polar body will be released, and egg changes into the ovum. So the pronuclei of both the gametes, already in the egg or the ovum, the nucleus is present, and another nucleus from the sperm is also entered into the egg, and both are fused. So here the both gametes are united and approach and have haploid number of chromosomes. So both the pronuclei have, nucleus of the gametes have the haploid number of chromosomes, and chromosomes of both nuclei come together and arrange on the spindle, and ovum with diploid number of chromosomes is called as zygote. So this is the This after combining the haploid and haploid male gamete and haploid female gamete, so the ovum is now with the diploid number of chromosomes, hence it is called as zygote. So next slide, in the diagram you are going to see the entry of sperm in the right side, entry of sperm into the egg, how the sperms are entering into the egg. So by dissolving the plasma membrane and wetland membrane in the animal pole. So that you have to see. Okay. So this is the fusion of sperm and egg. So this diagram shows there is a first figure. So in that there is a animal pole and vegetal pole. The sperm is going to be enter. In the second diagram already it is making the entry very close to the egg, and it is making the entry into the egg. So after entry, what happens, the fusion of both the pronuclear takes place, and diploid zygote is going to be formed. So this is the diagram. So this is about the fertilization, how the fertilization takes place in the frog, or frog ಹೆಂಗೆ ಹೆಂಗೆ ಆಗ್ತದೆ ಫರ್ಟಿಲೈಸೇಷನ್ ಅದೆಲ್ಲ ಗೊತ್ತಾಯ್ತು ಈಗ. ಆ್ಯಂಡ್ next is cleavage. So after fertilization, zygote will be formed, and the zygote undergoes rapid repeated mitotic division, and that forms the number of cells called as blastomeres, and this is called as cleavage. So what is cleavage? Cleavage is the rapid repeated mitotic division of the zygote. So the zygote resulting in the formation number of cells called as blastomeres. So here in the frog, the entire egg divides, and hence the cleavage is called as holoblastic cleavage, because the complete egg divides, hence it is called as holoblastic cleavage. Next, here in the blastomeres are dissimilar in size, and so the cleavage is unequal. Because if the both the blastomeres are similar in size, then it is called as equal cleavage, because they are equal. In the frog, the blastomeres are dissimilar in size, hence it is called as unequal cleavage. So there are in the first cleavage, it is meridional, means it passes through the egg center, it resulting in the formation of two blastomeres, two equal blastomeres. So it is the first one. ಅಂದ್ರೆ ಫಸ್ಟ್ ಕ್ಲಿವೇಜ್ನಾಗ ಏನಾಗ್ತದೆ ಅಂದರೆ ದ ಎಗ್ ಡಿವೈಡ್ ಆಗ್ತದೆ, ಆ್ಯಂಡ್ ಮೆರಿಡಿಯೋನಲ್ ಅಂತ ಕರಿತೀವಿ. ಮೀನ್ಸ್ ದ ಪ್ಲೇನ್ of first cleavage is meridional. It passes through the egg center. It resulting in the two equal blastomeres. Okay. And second one is, it is also vertical but at right angles to the first one, and resulting in the formation of four blastomeres. Then the second cleavage, what happens? It is a meridional cleavage or vertical cleavage, and it will be the right angles to first one, and it divides the first two blastomeres and produces four cells. So this is the second cleavage. And the third cleavage is latitudinal and occurring slightly just above the equator, that is towards the animal hemisphere. So this results in the formation or production of the 8 blastomeres, of which 4 are smaller and another 4 are larger. The third cleavage is horizontal, or we can call it as latitudinal cleavage, and resulting in the 8 celled stage that formed that cell stage is going to be formed and having two tiers or arranged in two tiers, that is 4 in the upper hemisphere, means the animal hemisphere, and 4 cells in the vegetal hemisphere. So hence they are arranged in two tiers. So the smaller blastomeres are situated in the animal hemisphere and they are called as micromeres. The larger ones are the larger cells or the blastomeres are called macromeres and are located at the vegetal hemisphere. So this you have to remember in the third cleavage. And the fourth cleavage is double, means the furrows are meridional and this produces 16 blastomeres, 8 in animal pole and 8 in the vegetal pole. So smaller cells which are present in the animal cells are called micromeres and the larger ones are present in the vegetal hemisphere, they are called as macromeres. The fourth cleavage is also double. Then the fifth cleavage is also double, means the furrows are latitudinal. So one bisects the micromeres in the animal hemisphere and the other cuts the macromeres in the vegetal hemisphere. So in the next slide the diagrams are there, you just observe, that is the diagrams of the cleavage, how the cleavage takes place in the frog. So in the fifth cleavage what happens? So one bisects the micromeres in the animal hemisphere and the other cuts the macromeres in the vegetal hemisphere. So this is a simultaneously two divisions takes place, that is double cleavage. The two planes will going to be divide the egg. So that is a latitudinal divisions. So this results in 32 blastomeres after the fifth cleavage. So 32 blastomeres are being arranged in the 4 tiers. So of these 2 tiers are micromeres and another 2 tiers are macromeres. So from this stage the regularity of the cleavage is lost. After the fifth cleavage, regularity of the cleavage will be lost and the blastomeres divide at differential speeds. And the smaller ones are going to be divide faster than the larger ones. So these are the processes taking place in the cleavage: first cleavage, second cleavage, third cleavage, fourth cleavage, fifth cleavage. So after fifth cleavage the regularity will be lost and the smaller ones are divide faster than the larger ones. So this is the diagram you are seeing in the slide. Okay, so next slide. So this is the diagrams. So cleavage diagrams. The first one is the egg and there is another cleavage diagrams as you are seeing in the diagrams. Next, blastulation. So after the cleavage the blastulation process takes place. Blastulation means it is the development of the blastula from the cleavage. Means the development of the blastula from the cleavage only the blastula development takes place, hence it is called as blastulation. So a solid ball called as marula is formed after the cleavage. So cleavage is after the cleavage there is a marula, a small stage, solid ball stage in as we have studied in the development of human. So here also after the cleavage there is a solid ball structure will be going to form, that is called as marula. At 8 to 16 celled stage the blastocele or segmentation cavity going to be appear. So blastocele increases by secretion into it an albuminous material from the surrounding cells and absorption of water from the outside. So here the formation of hollow spherical embryo, that is called as blastula, known as blastoderm, and that is the wall of the blastula. So as the cleavage proceeds, so here is it, as a cleavage, after the cleavage proceeds, a small cavity develops among the blastomeres, means the small cavity will be going to form in the blastomeres. So this cavity begins to develop in the 8 cell stage itself. So it is called blastocele or the segmentation cavity. So the surrounding blastomeres or the cells secretes a fluid that is called as albuminous fluid into it. So this fluid is known as blastocele jelly. So that blastocele jelly is filled is present in the cavity, or the cavity is filled with the blastocele jelly. The blastocele gradually increases in its volume as more and more blastocele jelly is poured into it. So this stage of the embryo is called as blastula. So as the blastocele increases in volume, so blastocele is going to be increases in volume, so the blastomeres arrange themselves into layers around the blastocele. So these layers are called, or this layer is called blastoderm, means the arrangement of cells, the wall of the blastula, so is known as blastoderm. So the blastoderm remains 2 cell thick at the animal pole, 2 cell thick in the animal pole. The sides and floors of the blastocele are formed of many layers. So the blastula of frog is called coeloblastula, because cele means cavity. So the in blastula there is a cavity is present, so that's why it is called as the blastula of the frog is called as coeloblastula, because it is hollow sphere. So this blastocele is not exactly at the center, hence it is called as eccentric, because see you are seeing in the diagram that it is not exactly at the center. So animal pole, vegetal pole, and the blastocele, there are 3 parts you are seeing here. So it is not exactly at the central position, hence it is eccentric and correct. So next, here you can observe clearly that is a zygote, already it has formed by the union of gametes, and after the zygote the cleavage takes place. So 8 celled stage, that is a cleavage, then the blastula is going to be formed, hollow ball of cells, and after the blastula, so gastrulation process is going to see now. So before the gastrulation there is a one more concept in the middle, that is called as fate map. So what is this fate map? Fate map means a chart showing the end result of the different areas of an embryo. What is fate map? The fate map means a chart that's showing the end results of the different areas of the embryo. So this is called as fate map. So towards the end of the cleavage, means ಯಾವಾಗ ಎಂಡ್ ಆಗ್ತಾ ಇರ್ತದೆ ಕ್ಲೀವೇಜು, towards the end of the cleavage the organ forming areas are well marked. So in the diagram you are going to see that is some areas. So archenteron, ectoderm, spinal cord, notochord, gut. So these all organ forming areas are well marked at the end of the cleavage. So here in the fate map, the macromeres of the vegetal pole, because the macromeres are present in the vegetal pole, so the macromeres of the vegetal pole develop into the endoderm, because the 3 germ layers has to be formed. So from the 3 germ layers only the organs are formed: endoderm, ectoderm, and mesoderm. These are the 3 germ layers. From these 3 germ layers only organs are formed. So hence the macromeres of the vegetal pole develop into the endoderm, means it is germ layer. Endoderm is a germ layer. So this germ layer is formed from the macromeres which are present in the vegetal pole. So next, the notochordal areas lies in front of the macromeres. So notochordal area. So notochord, in the main feature of the chordates, that is the presence of notochord. So this notochord area lies in front of the macromeres. So in between the endoderm and the notochordal area lies another area that is called as precaudal area. Precaudal area. So on the other side of the precaudal material lies the another germ layer called as mesoderm. Mesoderm is present. So in the next diagram you look the diagrams. So in the diagram you are going to see the nervous system, notochord, that is mesoderm, endoderm. So these are the germ layers, that is epidermis, neural tube stage, that is the transverse section of the neural tube, fate map of the embryo. Fate map means already I have told that in different areas, a chart which showing the end result of the different areas of the embryo. So in the different colors it has shown in the figure, that is central nervous system, notochord, mesoderm, endoderm, the different areas which have been marked. So the macromeres is formed the endoderm. Okay, the micromeres of the animal pole. So coming to the animal pole, the micromeres of the animal pole develop into another germ layer called as ectoderm. Okay, ectoderm is going to be formed by the micromeres present in the animal pole. So these micromeres present adjacent to the notochord and mesoderm and develop into a neuroectoderm. So the micromeres present away from it develop into the epidermal ectoderm. So the organ forming areas are well marked at the end of the cleavage. So these are the different areas are marked by the cells which are present in the cleavage, micromeres and macromeres. So next one is the gastrulation. So gastrulation process. So what is gastrulation? Gastrulation in frog. So gastrulation is the development of the gastrula from the blastula. So in the blastulation, what the definition says, blastulation is the process of formation of blastula from the marula or the cleavage. So that is called as blastulation. Like that, gastrulation means development of gastrula from the blastula, that is called as gastrulation. So the blastula has a single layer of cells, that is called as blastoderm. So only one layer of cells are present in the blastula, so that is a blastoderm. But in the gastrula it has three layers of cells, so that is endoderm, ectoderm and mesoderm. So during the gastrulation, the cells or the blastomeres arranged into three layers, not in one layer. They are arranged in three layers, that is ectoderm, endoderm and mesoderm. So these three layers are called as ಏನಂತ ಕರಿತೀವಿ ನಾವು germ layers ಅಂತ ಕರಿತೀವಿ. Here also in the slide you are seeing the migration of the various presumptive areas of single layered blastula to their proper positions in the gastrula. So the areas, presumptive areas, migration of various presumptive areas of the single layered blastula. Single layer blastula ಇದ್ದಿದ್ದು ಏನಾಗ್ತದೆ ಮುಂದೆ three germs layered gastrula ಆಗ್ತದೆ. Migration of the blastomeres take place ಅಂದರೆ morphogenetic movement ಅಂತ ಕರಿತೀವಿ. So here the cell division does not occur and the rearrangement of the cells to specific position occur in the gastrulation. So here during the gastrulation, what happens, the blastomeres move from one place to the another place. So these movements of cells during the gastrulation, movement of cells or the blastomeres during the gastrulation is called as morphogenetic movements. Morphogenetic movements. So the morphogenetic movements bring out a definite shape to the embryo. So these morphogenetic movements, so the resulting in the, or it will give the a definite shape to the embryo, hence the name. Morphological changes ಆಗ್ತವ, so it will give a definite shape to the embryo, hence the name is morphogenetic movements. So the morphogenetic movements are of two types, so namely epiboly and emboli. So in the diagram also you are seeing the different um, events which are occurring in the gastrulation process. So first see animal pole, vegetal pole, there is a egg diagram, and after the cleavage there is a blastola. Blastocele is present in the right side upper diagram, and gastrulation will be occur after the blastula. So here the different parts, dorsal lip of the blastophore, you are seeing, and archenteron is present in the gastrula, means the cavity present in the gastrula is called as archenteron. So in the blastula, the cavity present in the blastula is called as blastocele, but here in the gastrula, the cavity present in the gastrula is called as gastrocele. Otherwise another name for gastrocele is archenteron. There are three germ layers, that is ectoderm, mesoderm and endoderm. So there is a yolk plug is also present. So these are different events or different structures occurs during the gastrula development. So what is the mechanism involved? Already I have told that there are two types of cells in the morphogenetic movement. So one is epiboly and the emboli. So epiboly means the movement of the blastomeres, or the movement of the cells on the surface of the embryo. So that is called as epiboly. Epi means on the surface. The movement of the cells takes place on the surface of the embryo. That's why it is called as epiboly. So example for this is convergence, extensions. So these movements are the epibolic movements. And what is emboli? Emboli is the movement of cells into the embryo. So here emboli means emboli means inside the embryo the movement of the cell takes place, hence it is called as emboli. So in the epibolic movements, the examples are convergence and extensions. But in the emboli, the examples or the mechanisms involved in the emboli, or the examples for embolic movements are invagination, involution, divergence, etc. So these movements are called as embolic movements. So during the gastrulation, so some events occurs. So what are the events? So a groove appears below the gray crescent. The macromeres move into the embryo through this groove and form the endoderm. So the movement of endoderm into the embryo is called as invagination. So these are the events. Invagination ಅಂದ್ರೆ ಎಪಿಬೋಲಿ ಎಂಬೋಲಿಕ್ ಮೂಮೆಂಟ್ಸ್ ಇರ್ತಾವಲ್ಲ ಅವೇ ಕನ್ವರ್ಜೆನ್ಸ್, ಎಕ್ಸ್ಟೆನ್ಷನ್ಸ್, ಇನ್ವಲ್ಯೂಷನ್, ಇನ್ವ್ಯಾಜಿನೇಷನ್, ಡೈವರ್ಜೆನ್ಸ್, ಇವೆಲ್ಲ ಮೆಕ್ಯಾನಿಸಮ್ಸ್. ಸೊ ದೀಸ್ ಮೆಕ್ಯಾನಿಸಮ್ಸ್ ಯು ಹ್ಯಾವ್ ಟು explain. So the groove appears below the gray crescent region and the macromeres move into the embryo through this groove and form the endoderm. So this movement of endoderm into the embryo is called as invagination. Okay, so the groove depends into the embryo to form a cavity. That cavity is called as archenteron. Archenteron or formation of archenteron. The crescentic groove formed by the invagination of macromeres that mark the beginning of the cavity, that is called as archenteron or gastrocele. The opening of the archenteron to the outside is called as blastopore. So in the last diagram you are seeing the dorsal lip of blastophore. So that is the opening of the archenteron or the cavity gastrocele opening to the outside. So gastrocele is or the archenteron is going to be open to the outside is called as blastopore. The blastopore has the 4 lips, so namely dorsal lip, ventral lip, and 2 lateral lips. One is dorsal lip and one is ventral lip, and 2 are lateral lip. Total 4 lips the blastopore has. So a mass of endodermal cells protrude out through the blastopore called as yolk plug. So in the last diagram towards the right side you are seeing the yolk plug. So that yolk plug is a mass of endodermal cells protrude out through the blastopore, that is called as yolk plug. Then what happens is the mesodermal cells, precaudal plate cells and notocaudal cells. So all these cells migrate towards the blastoporal lips. So all these cells migrate towards the blastopore lips. So this movement is called as convergence. ಏನಂತ ಕರಿತೀವಿ ನಾವು ಕನ್ವರ್ಜೆನ್ಸ್ ಅಂತ ಕರೀತಾರೆ. Convergence. So after reaching the blastoporal lips, so all these cells reach to the blastopore lip. So after reaching the blastoporal lips, what happens? These cells roll over the blastoporal lips and move into the embryo. Again, they can move into the embryo by rolling over the blastoporal lip. So this rolling movements is called as involution. So convergence is different, involution is different, divergence is different, convergence is different. All these are mechanisms or the events which are taking place in the or taking place during the gastrulation. So the involuted cells move away from the blastopore into the embryo. So that is called as, or that movement of the cell is called as divergence. Means the cells move away from the blastopore into the embryo, that is called as the divergence. So next, the precaudal cells and notocaudal cells are arranged as a plate-like structure in the mid-dorsal roof of the gastrocele. So the mesoderm is arranged as 2 sheets on either side of the notochord. So in the early stage what happens? The notochord and the mesoderm remains as one continuous sheet called as chorda mesoderm. So above the notochord, the neuroectoderm cells are arranged as a plate-like structure called as neural plate, and it is formed by the extension. So the epidermal ectoderm extends all over the embryo except the area of neural plate and blastopore. So in the diagram you are seeing the ectoderm, endoderm, and archenteron. So where is endoderm is situated, where the ectoderm is situated, where the cavity, that is gastrocele or archenteron, is situated, and where the blastopore is situated. That is you are seeing in the diagram. Okay. So next one is, so what is the blastopore? Opening up the gastrocele is known as blastopore. So already I told that the lateral lips, ventral lips, and elongation. And the embryo slowly elongates mesoderm and splits a sheet from endoderm. That is the elongation. And delamination means it starts dorsally and proceeds ventrally. So these are the gastrulation in frog. So first one is the figure first A shows the late blastula and figure B shows the early gastrula, and C shows the middle gastrula, and in the below diagram and the D diagram shows late gastrula, and the E diagram shows early development of the nervous system. And after the gastrulation the organogenesis starts, and nervous system is going to be formed, and all these things happens. So gastrulation, the diameter of the blastopore is go on decreasing. The gastrula rotates within the fertilization membrane. The mature gastrula hangiruttappa, that is oval in shape, and it has a cavity called gastrula or the archenteron, and the archenteron opens outside or to the exterior by a blastopore, and a mass of endodermal cells project out through the blastopore is called as yolk plug. So it consists of three layers, namely ectoderm, endoderm and mesoderm. Ectoderm is the outermost layer, endoderm is the innermost layer, and the middle one is the mesoderm. So the roof of the archenteron is formed by notochord. The ectoderm lying above the notochord is called as neuroectoderm. So this is the mature gastrula. That is a late gastrula, mature gastrula. And after the formation of three germ layers, next is neurogenesis. So what is the neurogenesis or the development of the neural tube and curriculum? Neurogenesis under the development of the neural tube. So in the gastrula, the presumptive material of nervous system lies on the mid dorsal line as a plate like structure called as neural plate or medullary plate. In the slide you can see neural plate gives rise to neural folds. First the neural plate is present in the where in the mid dorsal line as a plate like structure, that is a neural plate. That neural plate gives rise to the neural folds, and these neural folds going to form neural groove. So the neural folds meet in the middle line and form the neural tube with a cavity called as neurocele. So at the anterior end, neural tube opens to the exterior by the neuropore. Then it will communicates with the gastrula, that is archenteron, by neurinetric canal. So the narrow strips, the neural crest on the sides, give rise to dorsal root ganglia of the cranial and spinal nerves and sympathetic ganglia and suprarenal glands. So neurogenesis means what? The development of the neural tube. So in the gastrula, the presumptive material for nervous system lies in the mid dorsal line. Already you have seen in the last diagram, that is called a neural plate. So this neural plate extends from the dorsal lip of the blastopore to the anterior end. So soon the edges of the neural plate becomes thick and raised above as neural folds, and the neural folds are going to be formed from the neural plate. These neural folds are two sides, so they are continuous anteriorly to form the transverse neural folds. So these neural folds enclose a shallow groove called as neural groove. That is what the first point explains. Neural folds. The neural folds increase in the elevation and bend towards one another until their edges meet and fuse. So thus a tube is formed called as neural tube. So it encloses a canal called as neurocele. So that is what the second point explains. The fusion first starts just behind the anterior end. The region, what means anterior end means in the region of the future medulla, and forms here the fusion proceeds both anteriorly and posteriorly. So anteriorly the neural tube opens to the exterior for some time by anterior neuropore. So that is a third point, and anterior end neural tube opens to exterior by neuropore, and it becomes closed soon, and posteriorly the neural folds enclose the blastopore in such a way that the neurocele, what it will do, it will communicate with the gastrula or the archenteron through the blastopore. So the short narrow canal that is connecting the archenteron and the neurocele is called neurinetric canal. The later it disappears. So this is the neurogenesis under neuronetric canal under fourth point. Then after the neural folds have fused in the middle line, the neural tube separates. So after the neural folds have fused in the middle line, what happens? The neural tube is separates itself completely. So from overlying epidermis, the free edges of the epidermis fuse together so that the epidermis becomes continuous over neural tube. Okay, so the narrow strips, neural crest on the side give rise to the dorsal root ganglia, cranial and spinal or sympathetic ganglion, so suprarenal glands. So these are also formed. So in the diagram you are seeing that is ectoderm. From the ectoderm what happens? So that is arrow mark. So groove, neural plate ಆದಮೇಲೆ ಏನಾಗ್ತದೆ? ನ್ಯೂರಲ್ ಪ್ಲೇಟ್ ಗೋಯಿಂಗ್ ಟು ಫಾರ್ಮ್ ದಿ ನ್ಯೂರಲ್ ಫೋಲ್ಡ್ಸ್ ಫಾರ್ಮೇಷನ್ ಆಗ್ತದೆ. ನ್ಯೂರಲ್ ಪ್ಲೇಟು ನ್ಯೂರಲ್ ಫೋಲ್ಡ್ ಫಾರ್ಮೇಷನ್ ಆಗ್ತದೆ. ಆ್ಯಂಡ್ ನ್ಯೂರಲ್ ಫೋಲ್ಡ್ಸ್ ಏನು ಮಾಡ್ತಪ್ಪ ಅಂದ್ರೆ ನ್ಯೂರಲ್ ಗ್ರೂವ್ ಫಾರ್ಮೇಷನ್ ಮಾಡ್ತದೆ. ಈ ಗ್ರೂವ್ ಫಾರ್ಮೇಷನ್ ಆದಮೇಲೆ ಏನಾಗ್ತದೆ ಅಂತಂದರೆ ದಿ ಲಾಸ್ಟ್ ನ್ಯೂರೋಪೋರ್ ಆದಮೇಲೆ ಕೆನಲ್ ಫಾರ್ಮೇಷನ್ ಆಗ್ತದೆ ಒಂದು ಇಲ್ಲಿ ಒಳಗೆ. ಕೆನಲ್ ಫಾರ್ಮೇಷನ್ ಆದಮೇಲೆ ಸೊ ಇನ್ ದಿ ರೈಟ್ ಸೈಡ್ ಆಲ್ಸೋ ದೇ ಹ್ಯಾವ್ ಗಿವನ್ ಡಾರ್ಸಲ್, ವೆಂಟ್ರಲ್, ಮೈಕ್ರೋಫಿಲಮೆಂಟ್ಸ್, ಮ್ಯಾಕ್ರೋಫಿಲಮೆಂಟ್ಸ್. ಮೈಕ್ರೋಫಿಲಮೆಂಟ್ಸ್ ಅಂದರೆ ಎದಕ್ಕೆ ಅಂತೀವಿ, ಮ್ಯಾಕ್ರೋಫಿಲಮೆಂಟ್ಸ್ ಅಂದರೆ ಎದಕ್ಕೆ ಅಂತೀವಿ, ಡಾರ್ಸಲ್ ಸೈಡ್ ಅಂದ್ರೇನು, ವೆಂಟ್ರಲ್ ಸೈಡ್ ಅಂದ್ರೇನು, ಇದೆಲ್ಲ ತೋರಿಸೋಂಥ ಡೈಗ್ರಾಮ್ ಅದು. ಸೊ ಇಲ್ಲಿ ನೋಡಿರಿ ಡೈಗ್ರಾಮ್ನಾಗ. ಸೊ ನ್ಯೂರಲ್ ಪ್ಲೇಟ್ ಅಂದರೆ ಎದಕ್ಕೆ ಅಂತೀವಿ ನಾವು. ದೆನ್ ಪ್ಲೇನ್ ಆಫ್ ಟ್ರಾನ್ಸ್ಫರ್ ಸೆಕ್ಷನ್. ಸೊ ದೆನ್ ನ್ಯೂರಲ್ ಫೋಲ್ಡ್ಸ್. So clearly this slides indicates the how the development of neural tube takes place. Neural folds from the neural plate, the neural folds develop from the neural folds. There is a neural groove is going to form. From the neural groove there is a canal. Which canal it is? That canal is called as neuronetric canal or neurosil. Okay. So in this type the neural tube is going to form. So that is the neurulation. So as the neural tube is separated from the ectoderm, here you can see the last diagram. From the ectoderm, the neural tube is separated from the ectoderm. A certain number of loose cells are liberated from the neural folds in the space between the ectoderm and the neural tube. These cells arrange themselves as two longitudinal bands on the dorsolateral wall of the neural tube. So these cells constitute neural crest. So later the neural crest cells differentiated into the ganglia of the cranial and spinal nerves and melanophores. Melanophores are also called as chromatophores and adrenal medulla and visceral skeleton. So the anterior portion of the neural tube differentiates into the brain. Means the brain is differentiated by the anterior portion of the neural tube and the posterior part into the spinal cord. So here you can clearly understand that the anterior portion of the neural tube differentiate into the brain and the posterior part of the neural tube is going to form or differentiate into the spinal cord. The embryonic stage which is having the neural plate or the neural tube is called as neurula. Neurula ಅಂತ ಕರಿತೀವಿ. So here you can see the diagram. Dorsal view of the embryo. That is the neurulation process. Neural plate ಇರ್ತದೆ ಫಸ್ಟಿಗೆ. ನ್ಯೂರಲ್ ಪ್ಲೇಟ್ ಆದಮೇಲೆ ಮುಂದೆ ಏನಾಗ್ತದೆ, ನ್ಯೂರಲ್ ಗ್ರೂವ್ ಫಾರ್ಮೇಷನ್ ಆಗ್ತದೆ. ಆ ನ್ಯೂರಲ್ ಗ್ರೂವ್ ಆದಮೇಲೆ ಏನಾಗ್ತದೆ, ನ್ಯೂರಲ್ ಪ್ಲೇಟ್ ಇದ್ದಿದ್ದು ನ್ಯೂರಲ್ ಫೋಲ್ಡಿಂಗ್ಸ್ ಆಗ್ತಾವೆ. ಆ ಫೋಲ್ಡಿಂಗ್ಸ್ ಆದಮೇಲೆ ದೇರ್ ಇಸ್ ಎ ಮಿಡಲ್ ಲೈನ್ ಅದಲ್ಲ, ನ್ಯೂರಲ್ ಗ್ರೂವ್ ಅಂತ ಕರೀತಾರೆ. ಸೊ ದಿಸ್ ನ್ಯೂರಲ್ ಫೋಲ್ಡ್ ಮುಂದೆ ಏನಾಗ್ತದಪ್ಪ ಅಂತಂದರೆ ನ್ಯೂರಲ್ ಕ್ರೆಸ್ಟು, ಆಮೇಲೆ ಸೋಮಾಯ್ಡ್ಸ್ ಅಂತ ಕರಿತೀವಿ, ಆಮೇಲೆ ಲಾಸ್ಟಿಗೆ ನ್ಯೂರಲ್ ಟ್ಯೂಬ್ ಡೆವಲಪ್ಮೆಂಟ್ ಆಗ್ತದೆ. ಅಂದರೆ ಇಲ್ಲಿ ಫಿಗರ್ ಎ ಏನದಲ್ಲ ಅದು ದಟ್ ಈಸ್ 19 days ಬೇಕಾದ ಫಾರ್ಮೇಷನ್ ಆಗ್ಬೇಕಂದ್ರೆ. ಆಮೇಲೆ ಏನು ನ್ಯೂರಲ್ ಗ್ರೂವ್ ಅಂದರೆ ನ್ಯೂರಲ್ ಫೋಲ್ಡ್ ಫಾರ್ಮೇಷನ್ ಆಗಬೇಕಾಯ್ತು ಅಂದ್ರೆ ಇಟ್ ಈಸ್ 19 days ಬೇಕು. ದೆನ್ 20 days ರಿಕ್ವೈರ್ಡ್ ಫಾರ್ ದಿ ನ್ಯೂರಲ್ ಕ್ರೆಸ್ಟ್ ಆ್ಯಂಡ್ 26 days ರಿಕ್ವೈರ್ಡ್ ಫಾರ್ ದಿ neural tube formation, somatid formation. This is a diagram, neurulation ಅಂತ ಕರಿತೀವಿ. So this is the cycle which shows the development of frog from egg to an adult. First you see the sperm and the egg, and the egg fertilization is external. The ova and the sperm released into the water medium, and in the water medium only they can fuse together to form the zygote, diploid zygote, called as fertilization. So after the fertilization, a fertilized egg undergo the rapid metatic division called as cleavage, and here the first, second, third, fourth, fifth cleavage, and after the fifth cleavage the irregularity or regularity of the cleavage will be lost, and that is going to form the solid ball called as marula. So from the marula there is a hollow ball of cells, structure is called as blastula. In blastula we are going to see the blastocyst. So after the blastocyst, from the blastula there is a 3 germ layered structure will be developed, that is called as gastrulation. And from the gastrulation, so in the gastrula stage we are going to see 3 germ layers, ectoderm, endoderm, mesoderm. So these 3 layers form the different organs. So neural tube going to be formed, and that neural tube there is a cavity called as neurula, and that after the neurulation ಏನಾಗ್ತಪ್ಪ ಅಂದ್ರೆ ಮುಂದೆ ಎಗ್ ಏನಾಗುತ್ತೆ, ಹ್ಯಾಚಿಂಗ್ ಟೆಕ್ಸ್ ಪ್ಲೇಸ್, ಮುಂದೆ ಹ್ಯಾಚ್ ಆಗ್ತದೆ, ಆರ್ಗನೈಜೆನ್ಸಿಸ್ ಆದಮೇಲೆ ಹ್ಯಾಚ್ ಆಗ್ತದೆ, ಹ್ಯಾಚ್ ಆದಮೇಲೆ ದರ್ ಇಸ್ ಎ ಲಾರ್ವಾ ಡೆವಲಪ್ ಆಗ್ತದೆ. ಸೊ ದಟ್ ಲಾರ್ವಾ ಆರ್ 3 germ layers give rise to the larval stage called as tadpole larva, and this tadpole larva undergoes metamorphosis. Metamorphosis means transformation of larval form to the adult form. So during this process many structures are going to be developed, and some of the structures will becomes reduced. So gill slits are going to be absent after the, so when it is in the larval stage the gill slits are present, after becoming adult the gill slits are lost. So it is in the aquatic form when it was in the larval stage. After acquiring the adult stage, so it is adapted to the terrestrial, and hence it is called as amphibians. Amphi means dual. The frog belongs to the class Amphibia and phylum Chordata. So amphibians means they lead dual life. They live as well as on land and in the water. So metamorphosis into adult. Okay, this is the development of frog. If you have any doubts you can ask me. You have any doubts? There is no doubts? Okay, so this is a very important question. You have to read 10 marks question. It is explain the gastrulation in frog. They may ask for 10 marks, or explain the different events occurring during the gastrulation. So invagination, involution, convergence, divergence, so complete process you have to explain. That is the gastrulation in frog. Sometimes 5 marks question, blastulation in frog, they may ask for 5 marks. Or sometimes complete question, explain the development of frog. Then you have to write from egg to this complete up to gastrulation. So sometimes 2 marks question, what is blastula, what is gastrula, or what is archenteron, what is fate map, and what is neurogenesis, and all these things will be asked for 2 marks. So this is the complete information which I have given in this session. Okay, thank you. We will end up the session.